Believe it or not, you're looking at a picture of glow-in-the-dark cats. These photographs haven't been touched up. These cats really can glow in the dark. They're the work of South Korean scientists. How did they make this happen? Through a process known as genetic engineering. This process goes by many names. You might also hear it called gene cloning, gene splicing, recombinant DNA, or genetic modification. In this slideshow, we're going to take a look at how and why this process is done. So first of all, what exactly is genetic engineering? Well, simply taking DNA from one organism, combining DNA with another organism, and getting a combination of that DNA known as recombinant DNA. Now, why would we do this? Well, there's a lot of different reasons. But essentially, when you give an organism new DNA, you give them the ability to make a new protein, and that protein gives them a new trait. So by combining DNA from different organisms, we get organisms that have unique traits. For example, this cabbage has been given a gene from a scorpion to produce scorpion venom. This way, the cabbage can defend itself against insect pests. These eggs have been given cancer-fighting proteins through genetic engineering, and so people who eat these eggs have the ability to treat or fend off cancer. These plants have been given DNA that makes them grow faster and makes them more resistant to changes in weather. These bananas may soon be genetically engineered with vaccines, so instead of going to the doctor for your vaccine, you'll just have to eat a banana. These goats are pretty cool. They can actually produce milk that contains spider silk protein. And then that spider silk protein can be harvested by scientists and can be used to make things that require super strong proteins such as umbrellas and parachute cords. We've also got plants that have a unique ability through genetic engineering. These plants can absorb lots of groundwater contamination, so they can actually be used to clean up toxic sites. Now, how is this even possible? Well, despite the diversity of organisms out there, all organisms share the same universal genetic code. All living things speak the same genetic language, and that genetic language is made up of 4 letters, G, C, T, and A. So even though this sheep, this bacteria, and this boy has a different sequence of DNA bases, they still have the same 4 bases. And because those bases are the same in all living things, we can combine DNA from different living things. So let's take a look at an example of this. Bacteria have been turned into human insulin factories. Now, why would we want to do this? Well, people with diabetes often need to take insulin shots to regulate their blood sugar. Human insulin is hard to come by. It used to be that diabetic patients would take pig or cow insulin, but that didn't work very well with their immune systems. So what we've done now is we've genetically engineered bacteria to make human insulin for us. Now, why would we choose bacteria as our factories as opposed to, say, elephants? Well, for one thing, bacteria are very simple. They've got one big bacterial chromosome. Then they've also got these small loops of DNA known as plasmids. And as you'll see in a minute, that's going to be really important. Another great thing about bacteria is that they reproduce quickly. They can divide asexually in a matter of 20 minutes, as opposed to elephants, it takes 20 months to actually have a baby elephant. So because bacteria reproduce so quickly, they're an excellent source for genetic engineering experiments. So as we go through this process, there's a couple important tools. One is restriction enzymes. These are enzymes that act as molecular scissors. They essentially cut DNA at specific locations. Another important tool is another enzyme known as DNA ligase. And DNA ligase has the opposite job. Its job is to glue together different pieces of DNA. Now, a couple things about restriction enzymes. There are many different restriction enzymes out there, and each different restriction enzyme cuts at a unique DNA sequence. So scientists, depending on what gene they're looking to cut out, will use a particular restriction enzyme to do the job for them. Here's an example of how that works. This is a bacterial plasmid, and these markers actually indicate where a particular restriction enzyme would cut. So if a scientist was looking to cut out this particular piece of DNA, they would want to use the BAMH1 restriction enzyme. If, however, they were looking to cut out this particular piece of DNA, they'd need to use a combination of two different restriction enzymes, EcoR1 and PST1. So let's see how this all works together to make genetically engineered bacteria that can produce human insulin. The first two steps are these. We take our bacteria and we remove the plasmid, that circular piece of DNA. We also want to remove the gene of interest from our human cell. In this case, the gene of interest is the gene that produces the human insulin hormone. Keep in mind that human insulin is a protein. So by taking out this gene from human cells, we are taking out the ability to produce human insulin hormone. Once we've got those two things isolated, we're going to cut them both with the same restriction enzyme. So here you can see the restriction enzyme has cut the plasmid in this specific spot, and it's also cut the gene for human insulin. Now that we've got those both cut out, you can probably predict what we do now. We're going to combine them together, and we're going to do that with the help of DNA ligase. So ligase will actually glue the human insulin gene into the bacterial plasmid right here. And now that we've done this, now that we have DNA from two different sources, bacteria and human, this is known as recombinant DNA. Once we've got our plasmid, though, this is only the beginning. We need to make a lot more copies of this gene of interest if we're going to produce lots of human insulin. So we're going to take that plasmid and we're going to put it back into the bacterial cell. And this process is known as transformation. We're essentially transforming this bacteria with a new trait because we're giving it recombinant DNA. This bacteria now has the ability, thanks to this gene, to produce human insulin. The rest is straightforward. We let that bacteria reproduce. In a matter of 24 hours, we can have millions and millions of bacteria. And every single bacteria is a clone of this original cell. And every single one of these bacterial cells can produce human insulin. Now, we simply harvest the products. We would remove some of the human insulin, for example, from the bacteria, and we can use that to treat diabetics. This process can be also used in many other applications. For example, the enzymes that are used to stonewash genes, repair faulty hearts, clean up oil spills, or genetically engineer plants. All of these different applications use the same essential cloning process. And here you can see again the many different applications. Pest-resistant plants, bacteria that can clean up toxic waste, proteins for heart attack therapy, growth hormone for those with stunted growth disorders, human insulin that we talked about, and acid wash genes. In class, we'll take a look at how this process works and some other interesting applications. Make sure that you took good notes on this slideshow. You can do the reading that follows this, add a little bit onto your notes, and then don't forget to complete the poll.